body. PET is mainly used in the area of medical imaging for detecting or measuring changes in physiological activities like metabolism, blood flow, regional chemical composition and absorption, and therefore, also called a functional imaging technique. Since this PET technique uses radioactive materials, also known as a tracer or radiotracer, for imaging, it is generally categorized within the field of nuclear medicine. A tracer is injected into the body, which gets trapped within the tissues of interest. The unstable nucleus of radioligand emit positrons, which combine with neighboring electrons to produce gamma rays in the opposite direction at 180 degrees to each other. These gamma rays are detected by the ring of detector placed within the donut-shaped body of the scanner. The energy and location of these gamma rays are recorded and used by a computer program to reconstruct three-dimensional, 3D, images of tracer concentration within the body. In modern PET computed tomography scanners, PET images are often reconstructed with the aid of a computed tomography X-ray scan performed on the patient during the same session, in the same machine. Different tracers are used for various imaging purposes, depending on the target process within the body. For example, 18FFDG is commonly used to detect cancer, 18FNAF is widely used for detecting bone formation, and 150H20 is used to measure blood flow. One of the disadvantages of PET scanners is its operating cost too. It can cost up to $8.2 million to set up a complete PET scan unit, and well over $1 million to obtain approximately 3,000 scans per year, depending upon the local cost of living. PET is both a medical and research tool used in preclinical and clinical settings. It is used heavily in the imaging of tumors and the search for metastases within the field of clinical oncology, and for the clinical diagnosis of certain diffuse brain diseases such as those causing various types of dementias. PET is a valuable research tool to learn and enhance our knowledge of the normal human brain, heart function, and support drug development. PET is also used in preclinical studies using animals. It allows repeated investigations into the same subjects over time, where subjects can act as their own control and substantially reduces the numbers of animals required for a given study. This approach allows research studies to reduce the sample size needed while increasing the statistical quality of its results. Physiological processes lead to anatomical changes in the body. Since PET is capable of detecting biochemical processes as well as expression of some proteins, PET can provide molecular-level information much before any anatomic changes are visible. PET scanning does this by using radio-labeled molecular probes that have different rates of uptake depending on the type and function of tissue involved. Regional tracer uptake in various anatomic structures can be visualized and relatively quantified in terms of injected positron emitter within a PET scan. PET imaging is best performed using a dedicated PET scanner. It is also possible to acquire PET images using a conventional dual-head gamma camera fitted with a coincidence detector. The quality of gamma camera PET imaging is lower, and the scans take longer to acquire. However, this method allows a low-cost on-site solution to institutions with low PET scanning demand. An alternative would be to refer these patients to another center or relying on a visit by a mobile scanner. Alternative methods of medical imaging include single photon emission computed tomography, SPECT, X-ray computed tomography, CT, magnetic resonance imaging, MRI, and functional magnetic resonance imaging, fMRI, and ultrasound. SPECT is an imaging technique similar to PET that uses radioligands to detect molecules in the body. SPECT is less expensive and provides inferior image quality than PET. Oncology. Whole body PET scan using 18FFDG. The normal brain and kidneys are labeled, and radioactive urine from breakdown of the FDG is seen in the bladder. In addition, a large metastatic tumor mass from colon cancer is seen in the liver. PET scanning with the tracer fluorine 18-18F, fluorodeoxyglucose, FDG, called 18FFDG PET, is widely used in clinical oncology. FDG is a glucose analog that is taken up by glucose using cells and phosphorylated by hexokinase, whose mitochondrial form is significantly elevated in rapidly growing malignant tumors. Metabolic trapping of the radioactive glucose molecule allows the PET scan to be utilized. The concentrations of imaged FDG tracer indicate tissue metabolic activity as it corresponds to the regional glucose uptake. 18F-FDG is used to explore the possibility of cancer spreading to other body sites, cancer metastasis. These 18F-FDG PET scans for detecting cancer metastasis are the most common in standard medical care, representing 90% of current scans. The same tracer may also be used for the diagnosis of types of dementia. Less often, other radioactive tracers, usually but not always labeled with fluorine 18, are used to image the tissue concentration of different kinds of molecules of interest inside the body. A typical dose of FDG used in an oncological scan has an effective radiation dose of 7.6 msv3 because the hydroxyl group that is replaced by fluorine 18 to generate FDG is required for the next step in glucose metabolism in all cells. No further reactions occur in FDG. Furthermore, most tissues, with the notable exception of liver and kidneys, cannot remove the phosphate added by hexokinase. This means that FDG is trapped in any cell that takes it up until it decays, since phosphorylated sugars, due to their ionic charge, cannot exit from the cell. This results in intense radiolabeling of tissues with high glucose uptake, such as the normal brain, liver, kidneys, and most cancers, which have a higher glucose uptake than most normal tissue due to the Warburg effect. As a result, FDG PET can be used for diagnosis, staging, and monitoring treatment of cancers, particularly in Hodgkin's lymphoma 4, non-Hodgkin lymphoma 5, and lung cancer 6, 7, 8. A 2020 review of research on the use of PET for Hodgkin lymphoma found evidence that negative findings in interim PET scans are linked to higher overall survival and progression-free survival. However, the certainty of the available evidence was moderate for survival, and very low for progression-free survival 9. A few other isotopes and radiotracers are slowly being introduced into oncology for specific purposes. For example, 11C labeled metamidate, 11C metamidate, has been used to detect tumors of adrenocortical origin. 1011 also, FDOPA PET/CT, or F18 DOPA PET/CT, has proven to be a more sensitive alternative to finding and also localizing pheochromocytoma than the MIBG scan 12-13-14. Neuroimaging. Main article, Brain Positron Emission Tomography. PET scan of the human brain. Neurology. PET neuroimaging is based on an assumption that areas of high radioactivity are associated with brain activity. What is actually measured indirectly is the flow of blood to different parts of the brain, which is, in general, believed to be correlated, and has been measured using the tracer oxygen 15. Because of its 2-minute half-life, O15 must be piped directly from a medical cyclotron for such uses, which is difficult. Example, in practice, since the brain is normally a rapid user of glucose, and since brain pathologies such as Alzheimer's disease greatly decrease brain metabolism of both glucose and oxygen in tandem, standard 18F-FDG PET of the brain, which measures regional glucose use, may also be successfully used to differentiate Alzheimer's disease from other dementing processes, and also to make early diagnoses of Alzheimer's disease. The advantage of 18F-FDG PET for these uses is its much wider availability. Some radioactive tracers used for Alzheimer's are fluorbutypar F18, flutamidamol F18, and fluorbutabine F18, which are all used to detect amyloid beta plaques, a potential biomarker for Alzheimer's, in the brain. Examples: PET imaging with FDG can also be used for localization of seizure focus. A seizure focus will appear as hypometabolic during an interictal scan. Several radiotracers, i.e. radioligands, have been developed for PET that are ligands for specific neuroreceptor subtypes such as 1,1-C roclopride, 1,8F-phallipride and 1,8F-dismethoxyphallipride for dopamine D2/D3 receptors, 1,1-C MCN 5652 and 1,1-C DASB for serotonin transporters, 1,8F-methway for serotonin 5-HT1A receptors, 1,8F-nifin for nicotinic acetylcholine receptors or enzyme substrates. e.g. 6-FDOPA for the AADC body. PET is mainly used in the area of medical imaging for detecting or measuring changes in physiological activities like metabolism, blood flow, regional chemical composition and absorption, and therefore, also called a functional imaging technique. Since this PET technique uses radioactive materials, also known as a tracer or radiotracer, for imaging, it is generally categorized within the field of nuclear medicine. A tracer is injected into the body, which gets trapped within the tissues of interest. The unstable nucleus of radioligand emit positrons, which combine with neighboring electrons to produce gamma rays in the opposite direction at 180 degrees to each other. These gamma rays are detected by the ring of detector placed within the donut-shaped body of the scanner. The energy and location of these gamma rays are recorded and used by a computer program to reconstruct three-dimensional